All right, good afternoon, everyone. Thank you all for coming. This is sort of a special Board of Health meeting. The meeting is now officially open. Excuse me. And just to inform you that this will all be recorded audio-wise and visually. We usually have public comments, but what we're going to do is continue our meeting tomorrow afternoon, our regular meeting with all the usual agenda items, so we will defer public comments until that time tomorrow. However, you, of course, can ask questions about this presentation. As you all know, one of our major health problems on this island is tick-borne disease. And we're fortunate to have with us today Dr. Kevin Asfelt from MIT, who has an interesting way and proposal of which way we may help conquer this. There's always a lot of discussion about deer, and people always badger me saying, why don't you get rid of the deer? And the other people tell me, no, don't get rid of the deer. The real issue, if you really understand the biology of the disease, is the disease, Lyme disease and the other tick-borne diseases are actually carried by the white-footed mouse. So there's been a lot of activity in the last few years about focusing perhaps on the white-footed mouse. Sometime this year, for example, there will probably be introduced a vaccine of sorts for mice, which may or may not work, we will see. But I think the proposal that you'll hear about today is really quite interesting. So it's with my pleasure, Kevin, to introduce you, and welcome. He has been here before, and we're working along with this project, and you'll hear about how we're involved and what he plans to do.
All right. Thank you, Malcolm, and thank you all of you for coming here today. This proposal really began about 7 months ago, back in June, when a number of you were here, Danica. And the idea, of course, is to prevent tick-borne disease, and specifically to prevent tick-borne disease at its source as an ecological problem, ideally coming up with an ecological solution. But like any community problem, it needs a community-driven solution. And that's why I'm hoping that we can get guidance and advice from all of you today, because really this needs to be a community-driven project, not something led by scientists. So the proposal, bluntly, is to break the transmission cycle of the disease in the environment, specifically by immunizing the white-footed mice so that they are resistant to Lyme and potentially to other tick-borne diseases, thereby introducing immunity to most of the native mouse population. These mice would be genetically altered, but the introduced DNA would be from other white-footed mice, so they would still be 100% mouse. We're not throwing in genes from bacteria or jellyfish or anything like that. They would be genes from other mice. And in fact, specifically, they'd be genes from other mice that have been exposed to these pathogens or to tick bites and had developed immunity to them, because normally when mammals like ourselves are exposed to a pathogen, our immune systems actually evolve molecular solutions that can recognize that pathogen, and that's how we learned to overcome it, and then we're immune thenceforth. But we can't pass that immunity on to our children. What we're proposing here in the mice is to take the immunity that some mice have evolved and encode it in the genomes so that they can pass it on to their children. That, in a nutshell, is the proposal. But again, this must be completely open and community-driven science. That is, if you're proposing to alter the environment in any way, shape, or form, it must be for a good purpose. It must offer obvious benefits to everyone. Discussions have to come before experiments. So when we were here last June, the question was, is there at least enough interest in this from both the board and from the community for us to even bother beginning experiments in the lab? And the answer then seemed to be a yes, we're at least interested enough for you to begin experiments, and so we have since then. Safeguards need to be agreed upon early. So in this case, no This is why safeguards are important. So what might go wrong? Solicit, frankly, criticism from everyone involved as to the things that might go wrong, because we can't possibly anticipate everything. So we want to hear what you think. You're the people who live here. What might go wrong? Tell us. Tell everyone. And we need to then figure out a plan for dealing with it and making sure that it doesn't happen before anything gets done. Projects like this, frankly, should be done nonprofit. If there is any possible way to do it nonprofit, it should be done nonprofit, because we don't need to introduce the profit motive as a complication here. There needs to be independent monitoring and analysis. And that's because, in general, in science, the last person you want to test a hypothesis is the person who came up with it, because they're the single most biased person in the world. Similarly, people who come up with ideas for potentially bettering the world probably shouldn't be the people that actually run the experiments, because if they're collecting data, measuring how effective it is, and looking for side effects of things that might be going wrong, well, no matter how good your intentions, you're going to be somewhat biased. So we need independent monitoring of the project. And above all else, everything must be done in the open. That means the science, that means the public comment, that means the finances, everything. It will only move forwards if embraced by the community. And above all else, please, please, please do not stop anything you are currently doing to fight tick-borne disease. This is a potentially years-long project. It would be at least 7 years before any mice would be released on Nantucket, with many points where the community would have to say yes before we get anywhere near there. And in the meantime, tick-borne disease is real. It is here. Please, please continue your existing measures to stop it. So we now have a steering committee with several new members for the project. And this is an approach really suggested by Malcolm based on his own past experience. And in order to proceed, the steering committee must secure federal, state, and local approval, because again, this is an environmental alteration and there are regulations for this sort of thing. All efforts must be guided by local and national ecologists. And we need to start small and scale up. So the trials will be sequential. Start on one or more uninhabited islands first. See how it works. Look for things that might go wrong in the environment. Have the independent monitoring committee deliver a report to the citizens, who would then decide, is this something to potentially try on Nantucket? Note that nothing we are proposing would be anticipated to spread beyond whichever island it's introduced upon. You might have heard a lot about gene drive, which is something else that I work on. That is totally unconnected to this project. We are not proposing anything of the sort. It would be radically premature. We don't want anything of that sort. What I really like to emphasize is we don't have a great deal of experience with changing the environment deliberately. Ecosystems are incredibly complicated. And when it comes to engineering a really complex system, you have to acknowledge that you don't know what might go wrong. We don't know. And that means that above all else, when you're thinking about what to do, you want to make the smallest possible change that has a chance of solving the problem. And then even once you've come up with what you think that change might be, you do not roll it out all at once. You test it in the smallest possible area that can tell you whether or not it's safe. And then only if things look good, it looks like it works, it looks like there aren't any major problems, would you then think about scaling it up. So those are the two guidelines for eco-engineering in general that I think very much apply in this case. So again, what are we here to do? We're here to block this cycle from happening in the environment. So, of course, tick-borne disease is carried by ticks, and we get it when they bite us. But the ticks are not born infected. They get infected when they bite an infected, usually rodent, usually white-footed mouse. So the pathogens, such as the Borrelia burgdorferi, which is the spirochete bacterium that causes Lyme disease, the pathogen that causes Lyme disease typically gets into the tick when it bites an infected mouse. But mice are also not born infected. They get infected when they get bitten by an infected tick. So really, the pathogen persists in the environment, being passed back and forth between mice and ticks. And we wouldn't care except for the fact that the ticks bite us too. Now, the mice actually don't get disease when they get infected. They're more or less immune. They've been at it long enough, they've been exposed long enough that they've evolved a sort kind of truce. We don't have that luxury. So we get Lyme disease. But what would happen if the white-footed mice just couldn't become infected at all? Well, if the mice were immune, then when the ticks, they wouldn't get infected when bitten by infected ticks, and then they wouldn't infect new ticks. And so you'd have fewer infected ticks in the environment, and then when you get bitten by a tick, there'd be a much lower risk of getting disease. That's the idea. If every mouse produced antibodies, again, from other mice, that conferred effective immunity from birth, then you would break this cycle, and the reservoir of both the Lyme-causing pathogen and potentially, depending on how you do it, the other pathogens that cause tick-borne disease would likely collapse. In an ideal world, if you had no infected mice, then you would have no infected ticks and no infected people. In reality, it's more complicated than that. Some ticks could become infected from secondary reservoirs. The mice are not the only rodent out there. They're just the best one at infecting ticks and the best one at being infected by ticks. And the reason why there's a problem today, in large part, is that there are many more white-footed mice relative to the other things that ticks can bite. It's also true that you have more ticks because you have more deer, but above all else, more white-footed mice means more infected ticks because they are the best at infecting the ticks and they are the best at getting infected. So if we immunize the mice, it's not going to go away. It's going to go back to a level that is more historically reasonable before we had so many mice and so many ticks. So there are several different ways of going about this, and I'm going to outline the different options for ecological vaccination in general. Option 1, immunize the mice against the Lyme pathogen only. So this wouldn't affect any of the other diseases. It would only prevent Lyme disease. And this would be probably the most conservative intervention of this type. And I say that just because it would only target a particular molecule present on the Lyme pathogen. It shouldn't affect the ticks, it shouldn't affect anything else, just that pathogen. Option 2, immunize the mice against the ticks, specifically against the tick saliva. So tick saliva has things that prevents the immune system from attacking the tick when it bites you, because the tick has to remain attached for a long time, especially to transmit the pathogen. If we immunize the mouse against the tick saliva, then we break that protection, and the ticks can't feed for long enough to acquire a pathogen or to transmit a pathogen. So that would block not just Lyme, but also Babesia, Anaplasma, and Ehrlichia, the other main kinds of pathogen. However, it would also reduce the number of ticks in the environment, because again, the mice are the most abundant prey animal for the ticks on Nantucket. And if you immunize the mice against the ticks, you're going to have fewer ticks. That might be something you're okay with. But that might have ecological effects in a way that option 1 would not. Now, to be fair, there are many more ticks than, again, quote unquote, there should be, largely because there are many deer, and the deer population is much higher, and the two are linked. But it would go down. That might have effects. We don't know what they are. Option 3, just do both. If you're going to immunize the mice anyway, the tick population is too high, block Lyme as effectively as you can, and block the other ones as effectively as you can. And option 4, of course, is the status quo. Don't do anything like this. Look for other sorts of interventions. Throw everything else you have at it. I would personally encourage you to do that anyway. That is, again, this is something that would not move forwards until at least 7 years from now. Throw everything else at it in the meantime, in a concerted effort, everything else that you're comfortable with, and then decide, is that good enough? And if it's not, then consider this. If you haven't done that, then I don't see why you would consider this. So please, by all means, do that. How does it work? Well, so what we would do in the laboratory is we can deliberately expose the mice to either the Lyme pathogen molecule or to the tick saliva. So we vaccinate them and they become resistant. Specifically, they become resistant because they've evolved antibodies that recognize the molecule that we've exposed them to. We can then sequence the genes from their immune cells that have evolved those antibodies. And then we can make those antibodies in the laboratory from those antibody encoding genes. We can then pull out the ones that are the most protective. And then we can encode those particular genes into the mouse genome so that they express these native mouse antibodies from birth and can pass it on to baby mice. So that's really what's different about this proposal, is it's stable. Not stable in the long-term sense. Anything we do to the environment, nature will win in the end. That is, natural selection will win out. It might take it decades, it might take it centuries, but it will eventually return things to baseline. There is nothing we can do to stop that. We will be immunizing the mice here, but it's probably not going to create enough of a benefit to the mice to be stable. Again, they're not normally affected by most of these pathogens. So eventually it will go away. But eventually means decades to a few centuries in all likelihood. That is, long after we care, or if we need to sustain it, then that's fine. So I would view that as a plus. In the long run, everything we do is temporary. But again, we don't understand the ecosystem. So you need to start small, see what the effects would be, and then only scale up if things look good. So we would need a field trial in which we would release these mice, which would be initially engineered from a population in the laboratory, isolated from here, in fact, and then crossbred to the native mice here to generate a population that is genetically identical to the mice that are already here, or in this case on the uninhabited island, and then released. And then you would have the independent study of the effects, looking at what happens to the mice, what happens to the ticks, what happens to the pathogens, what happens to the things that eat the mice. Does anything go wrong with the ecosystem as far as we can tell? And again, this would need to be independent. And then that independent committee would then deliver a report to the citizens here. And you would have some form of, we'll get into details later, but effectively a vote. And then you would have trial 2, which would be the real thing potentially here. And again, the timeline is potentially, this is 7 years from now, at the earliest. That's if we're really good in the lab. So again, here's the timeline. We're giving ourselves 2 or so years to identify the most protective antibodies of each kind and to engineer mice that are as resistant as we can make them in the lab. That might take longer. It's an engineering challenge, but biology is complicated. Once we have them, then we need enough mice to release onto a small uninhabited island to constitute a significant fraction of the mouse population on that island. We need most of the mice to be immune. So it takes time to breed mice, roughly 3 years. Then once we have released them on an uninhabited island, it's at least 2 years to evaluate the effects. And that's because the tick generation time is 2 years. So if you want to see what happens to the fraction of ticks that are infected, then you need to wait at least 1 full tick generation. And only then would you consider a release on some place like Nantucket. How would that actually work? Well, you release immune mice over 1 to possibly 2 years. The mice would increase the local population to at most 200% of normal. That is, ideally you release about as many mice as are currently here, ideally at the low point in the mouse population cycle. That is, at the end of winter. Say a month or so from now would be ideal. But for context, mouse populations will typically fluctuate by about 400% or more. Sam Telford is probably the leading ecologist on this, who's studied it in this particular cycle for 30 years, estimates that you get up to an 8-fold change in the mouse population over the course of the year. So we'd be releasing a goodly number of mice, but it would not go to the high that you see at the end of the year by any means. And if that is a problem, if that just seems too many mice get in your houses, mice potentially cause problems, then by all means you could lay out bait stations and traps to reduce populations near areas that you care about. That would not affect the overall project. What you care about is the balance of mice that have the resistance genes put into them from other mice. So the status, again, this really began back in June of last year, and we've since also talked with various agents on the vineyard. I have to say it's much easier to talk to all of you, just because working with Malcolm in particular and Roberto has been a delight, everything moves very smoothly and is well organized. It's delightful coming out here. There we're working through. We have the Board of Health here agreed to jointly develop a project management plan, largely designed by Malcolm. And in December, they nominated Dr. Emily Goldstein Murphy to join the steering committee for Nantucket. On the vineyard, we have 4 out of the 6 towns have agreed to send nominees to the committee. 2 more, we need to give additional presentations to those particular towns and boards of health. And in the laboratory, our collaborators have finished vaccinating white-footed mice. We have all our bureaucratic paperwork done to raise mice at MIT, and we've had the white-footed mouse genome sequenced. And that's just important because we need to know where can we put in the antibodies without causing something else to go wrong with the mice. We need to know what we're looking at. And we've been awarded a grant from the Greenwall Foundation to support our ethicist, Jantine Lunshoff, to essentially keep an eye on us throughout the project and ideally figure out what should a project like this look like from an ethical standpoint. Because we have a lot of work on informed consent in medical research. Do you consent to a treatment in medicine? But that's very different from a treatment in the ecosystem of an entire community. So how do we go about this? We're still figuring this out, and we need your help. So again, we have the steering committee will be about 5 to 7 members, ideally, including the Board of Health representatives. We would have what we're calling a data safety monitoring board, and that's the term from, again, medicine. There isn't really an equivalent for ecological projects like this. So we're trying to pull everything that is useful and relevant from that area and really anywhere else. And we're working on exactly how big that would be. The current best idea is go to the National Science Foundation and invite them to appoint unbiased people. But possibly you would want to voice in particular people on that as well, in terms of locals, which would be great. And then we have our project manager, who is Joanna, who's a member of our group. She's actually, I'm afraid, from the vineyard, but she is wonderful, and you should talk to her. Decision points. There are obviously a lot of these. If you tell us to stop at any point, then we stop. That's it. No project like this could ever go forward without community support. Just must never happen. But there are 2 main decision points. The first one is after we've made the mice in the lab. We've made them as resistant as we can, and then the Data Safety Monitoring Board needs to look at our data, saying how resistant are these mice? Are they in fact mice that are identical to the mice that are already there, except that they have these extra antibodies isolated from the mice that have been vaccinated? And they would deliver a recommendation to you and to the citizens of the vineyard. And if the recommendation is go, then we would need a regulatory approval for the next phase. So federal and state primarily, but also approval for the use of an uninhabited island. Again, we're still looking for what would be the best island. Obviously, there are different considerations, private versus public, different regulations, and it needs to be appropriate from an ecological sense to be a good candidate that would let us model and figure out, is this actually what it would probably look like on Nantucket? And we would, of course, need continued interest from the Board of Health to move forwards. After that trial had been completed, so again, you're looking at least 2 years later, you would then have the Data Safety Monitoring Board would look at all of the data collected on that island, either by them or by other investigators, but they would do all of the analysis, and they would deliver a report, and specifically a recommendation. Is this effective, and does this appear to be safe in the environment? If so, then we would need regulatory approval for the next phase, again, and a warrant proposed by the Board of Health at a town meeting, and warrant approval at a town meeting in order to begin releasing mice here on Nantucket. So again, at least 7 years from now, but that is our timeline and progression. Again, I've said all funding has to be open, so where's it coming from now? Well, our laboratory at MIT is now 1 year old, so we're mainly relying on initial support from MIT and some from Tufts, where our collaborators are at. And we have the Greenwall Foundation grant. We've applied for some government grants for development of the immune mice in the laboratory. And then we would need to raise money from presumably philanthropic sources, probably multiple sources, for scale up to the uninhabited island field trial, and then eventually for releases on Nantucket and the Vineyard. And again, whatever that funding comes from, it has to be in the open. I'd even be leery of an anonymous donor, frankly. Yeah. And it's fair to say that other projects of comparable scale probably require funding in the millions, just given for the monitoring and safety. But again, we're hoping that we could raise it from philanthropic sources. So here are the big questions for all of you. That is, first and most, is this project still one that you might wish to pursue? We're not asking you for approval to go ahead. Again, there's many, many points where you will have to continue to say yes to this question. But at this time, is this one you're still interested enough that poor Joanna should actually continue working with mice in the lab? If so, then what should we do? Should the mice be immunized against the Lyme pathogen specifically, or against tick saliva, or both? And given that there are options and we can obtain approval, where should the first field trials be held? Some people have suggested Tuckernuck as a potential candidate. What criteria must be met for you to consider releasing mice on Nantucket eventually? Again, you have years to answer this question, but the earlier it's put out there, then the better the discussion the community can have. And lastly, do you want to nominate anyone for the steering committee? Please let your Board of Health representatives know. And we also have a few questions that have been submitted by local residents that I would just like to address before we open it up in general. So would this amount to wiping out the existing mice and replacing them with a new species? So no. Again, these mice would be from the same native population. That is, we'd take the mice from the lab, which are native to, I think, are they Vineyard or Nantucket? I will need to ask Sam Telford. But they would be crossbred to local Nantucket mice for at least 8 generations. So they would be genetically identical to the native Nantucket mice before they would be released, except for the introduction of the antibodies from the vaccinated mice. And in fact, the State Fish and Wildlife, we had a meeting about a year ago before we came to Nantucket, just to sort of with the regulators and ethicists and representatives from environmental NGOs active out here on the islands, just saying, okay, what would we need to do if this project were to go forwards? What is the best way to go about it? And we had the head of State Fish and Wildlife, Tom French, there, and he said, well, above all else, from our point of view, you can't release any mice that if they're any different from the ones that are already there. So that's what we have to do anyway. There's a question about have any of the pathogens spread by ticks in this area, have they been genetically modified in any way? And the answer is several of them, yes, in the laboratory, but they're actually kind of a pain. We haven't done it ourselves, but I've talked with many people who have. I just went to the main Lyme conference a couple months ago, and they are surprisingly difficult to edit. But those engineered lab pathogens, all they've done is, say, drop in an antibiotic resistance gene or a fluorescent gene so that they can see that it's there. They haven't done any substantial work. And that's largely because they're a surprising pain to work with. You might have heard of a tool called CRISPR that makes it very easy to edit things. Even that doesn't make this particularly easy. And I'm happy to share the available literature on what we know in terms of that. So if this works, what would stop the pathogens from just moving to other organisms, to birds, reptiles, dogs, cats, horses, and so on? And the answer is they're already there. All of these animals get infected when they're bitten by ticks. They're not necessarily stably infected. Some of them get sick. Dogs and cats do, just like we do. But some of them don't, or at least not as much. But the reality is that for all of them, because there's fewer of them that get infected, fewer of them that stay infected, fewer of them that are capable of infecting uninfected ticks and thereby continuing the cycle, that if there are fewer ticks that are infected by the mice, which infect the majority of them, then that means fewer infections of all these other animals in the next generation, because you'll just have fewer infected ticks, which means fewer infected ticks biting these other animals. Fewer of them will be infected. To the extent that they can infect ticks, you'll have even fewer infected ticks in the next generation. So to some extent, the deck is stacked against the evaluation of this, because we'd be looking after one tick generation. but you would expect it to keep going down. But we're not going to see that. What about co-infection? So here's one of those cases where biology is complicated. There's been a lot of studies of what happens if you have a tick that is infected with both the Lyme pathogen and, say, Borrelia. Can you get both when the tick bites you? And the answer seems to be, well, yes, you absolutely can, and that's really terrible. But what happens if the presence of both of them make it easier for both of them? Does transmission increase? And the data, frankly, is not very good. The answer is probably yes. But again, that's one potential reason to go for the anti-tick approach, because that would, by disrupting tick feeding, it blocks all of them, except for the viruses, which are absolutely there, but they're very, very rare compared to the others. So you should have fewer infections of all kinds of these, including the co-infections. So we don't think that the unknowns on co-infections should affect this. And lastly, again, how are you going to test the effects without potentially causing irreversible damage? It's all very well and good to say uninhabited island as if no one will be affected, but of course all the organisms living on that island would be affected. So if something goes wrong, then what are the plans for doing something about it if the independent monitoring committee sees that something goes wrong? Part of the reason why they're there is to notice that and call for a halt to the trial and for it to be reversed insofar as is possible. So that's what happens in medicine with clinical trials, and that's what should happen here. So how do you reverse it? Well, in this case, you just release a lot of wild-type mice. Same principle in reverse, and you just dilute out that gene as fast as you can. You set out a lot of traps for the mice that are already there that would have the anti-Lyme, anti-tick antibodies, and you would release a lot of native wild-type mice, and you just dilute it out. And on a small uninhabited island, that's not that many mice. That is, we could trap mice on the mainland or here or the vineyard, as long as we're careful about which subspecies is where, and just move those over to the island and dilute it out as quickly as possible. Again, if you build a dam, the environment will change. If you remove the dam, it will not necessarily go back to the way it was before. Usually it does, but not always. That is true for any kind of environmental change. So in this case, we could switch it back very quickly, in less than one full tick generation. But there are no guarantees in life, in any kind of complex system, and that's all of biology. And that's why, even though all changes we make have risks, there is always risk in life, there's also risk in doing nothing, but the goal is to make the smallest possible change capable of solving the problem. And my suspicion is that adding immunity genes from the mice that happen to be immune already to make all the mice immune is probably the smallest change that looks possible that we could actually accomplish. Again, here's our team. We're very, very grateful, in particular to Sam Telford, who's been doing tick ecology on Nantucket for a very long time. Lyndon Hu is one of the leaders in Lyme from the medical perspective as well, is also on immunization. And Ryan and Aurelie are a senior grad student and a postdoc helping us out from their groups in a collaborating group. And then here is our research team, and then John Teen, our collaborating ethicist, keeping us in line. But we're also very grateful to everyone else in the Board of Health, and we're very happy to take any and all questions, not just in this format. If you have a question for one of us that you don't want to raise in public, please just email us. Happy to respond directly. But please, anything that you think that you're concerned about, anything that you're hopeful for, anything that you want to know more about, please let us know. That's what this is for.
If you could come down, come down to the microphone so we can all hear you.
Could you explain a little more about how, starting with half modified mice, half normal mice, it spreads to the whole population? Is that because it's a dominant characteristic where you only need one copy, so a mating will produce an immune mouse?
Yes, that's exactly right. You only need one copy of the antibody encoding cassette to produce enough antibodies to be resistant. Again, assuming that we can do that. And ideally, this is getting a little complicated, but we would want to insert multiple copies of that identical cassette around the genome so that you only need one to be immune, but the initial mice we release would have a lot. But when they have babies, then the babies on average would have half as many. But then that means that every copy in that first mouse is sort of like releasing an additional mouse without having to do that. So the goal would be to release mice, I mean, ideally if we could, with 100 copies each, and then it would diffuse out among the population. We'd have to release very few mice initially. But we don't know how many copies we can insert at a single step. There's a feasibility of breeding them up. It gets dilution, complicated genetics. But yes, you need one copy. And so if we release a number equivalent to the existing population, even if they only have 2 copies each, that statistically should result in most of the mice being resistant.
Have you considered how this might affect the predatory food chain, such as hawks and the feed on the mice and that whole cycle?
Yeah, great question. So that's, again, when it comes to what interacts with what in the environment, again, the things that eat the mice would be the most direct. So the change in this mice is the mice would be producing antibodies that are probably present in some other mice anyway. So you're not adding any new class of molecule that is different to the mice. They might, what's different, I suppose you could say, is that in the baby mice, they would have antibodies, when the baby mice wouldn't normally have antibodies. So you might imagine that a predator that only eats baby mice might have some sort of reaction, but it's pretty darn implausible. Again, that's why start small and scale up. Monitoring the predators would absolutely be something that would have to be done. People have raised concerns, well, what would happen to my cat if it eats one of these mice? And that is a study that we can absolutely do in the lab. We absolutely have cats that would be delighted to eat lots of mice.
Once you genetically modify a mouse and release it in the environment, will that trait be passed on forever, or does it have to be reinforced?
Great question. So whenever we alter an organism in essentially any way, we are reducing its ability to survive and reproduce in the wild. Because nature is better at that than we are. Right? That's what evolution is optimizing out there every day. And the odds that we can improve upon that are vanishingly small. So whatever we do, it will be passed on, but the odds are that those mice are less likely to survive to reproduce than normal mice. How much less is going to depend on the nature of what we're doing to them. In this case, hopefully it's not too much of a penalty because we don't want it to go away sooner than, a couple of decades. But it will eventually go away. The question is decades, centuries.
So nature will take care of this change and revert back to what was normal before the experiment.
Almost certainly, because the mouse is expending energy making these antibodies, which do very little for the mouse. So that's a burden. It's a tiny burden compared to a whole mouse making a few tiny molecules, but it's still a burden. It's still wasting the mouse's resources that could be better spent doing something else.
So to be effective, you have to keep introducing modified mice to the environment.
My guesstimate is you might want to check back in 20 or 50 years and see how it's doing.
All right, I don't have that kind of time.
But, yeah.
I don't think any of us do. Well, in 20 or 50 years, I'm sure we might even have human vaccines by then, so who knows? If we're lucky.
One comment and a question. The comment is, being a Pollyanna, I would definitely favor going after the tick because we get rid of all the tick-borne diseases. And it seems that developing an attack against the tick saliva would mean going after just the fibrinolytic enzymes that are not letting the blood clot and therefore making the bite good. So it would be a very specific thing. You wouldn't be going after an antigen which is sort of pleomorphic and you could have several different antibodies against it. You'd be going after one thing and we'd be getting rid of everything in the diseases, Lyme, the babesiosis or ehrlichiosis, and so forth. The question is, how is gene drive less specific than what you're proposing to do? It would seem to me that it would make the whole project more powerful. You'd need fewer mice, and unless there's some question of lateral transmission of something that I just plain am not seeing, I'd really appreciate hearing your thoughts on that.
All right, so thank you. This does require me to explain gene drive. So normally you have 2 copies of most genes, which means that if you have 2 different versions, or suppose you have 1 chromosome that has the antibody cassette in this case, and 1 that does not, then that means that if your mate has 0 copies, then that means that the offspring have a 50% chance of getting the one that does. This is how most genetics works in sexually reproducing species. A gene drive is a system that biases inheritance in its favor. So if you attach a gene drive to that antibody encoding cassette, then it would be almost guaranteed to be inherited at the expense of the unaltered one. And this could therefore spread immunity through a population much more quickly. You could release many fewer mice. It would be much more cost effective. In a sense, it would be a smaller change because you could release fewer mice in order to initiate it. The problem is the easiest way to build a gene drive right now biases inheritance in the first generation and the second and the third and the fourth and so on ad infinitum until eventually you've altered all of them. That is, not necessarily all of them just on an uninhabited island or on Nantucket, but as soon as you get even 1 to the mainland that reproduces, it keeps going. Now, this is lovely, potentially, if we're sure that this is exactly what we want to do. But again, rule 2 is you start small and local, and you see if it works, see if anything goes wrong, and you only scale up if warranted. So that form of gene drive we call a global drive system because you have to assume that it will spread around the world. So it violates rule 2 about as profoundly as it can possibly be violated. It is a self-scaling system. If you build and release 1 anywhere into the wild in the world, and it's lucky enough to get established, you're probably not stopping it unless you build another one to remove it. And again, this is an active area of development for our group. This is not related to the project. And unfortunately, if you Google my name, you will come up with lots of gene drive because that's the other thing we do. But I do not recommend anything like a gene drive system for projects like this. It would be tremendously premature. We definitely don't want it to spread forever. I think people would be very unhappy were it to spread to their community when they were not consulted in these discussions. We are working on forms of gene drive that will not work every generation, that effectively have genetic fuel and they will run out. So they will copy cells for 6 generations and then stop. Those are still in development in the laboratory. Maybe 7 years from now we'll have something decent. It would be an option. In the event that we do, and we've demonstrated that we think it might be safe enough, then we might propose it to you at that point, but probably only if people like you say, hey, we really, 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 really want this. Because this is a project that does not require gene drive. And as a general rule, when it comes to very new technologies like that, if it's not absolutely required, don't take the risk.
Thank you for your willingness to provide all this information, and I have a whole bunch of questions, so if you
Please.
bear with me. First, of all of the work that you're planning on doing in this project, have any parts of it already been done and proven to work in some small way with different species, different goal? But is any of this already proven?
That is a great question. So we are in many ways, you might call it cherry pickers. That is, a lot of great basic scientists have done a lot of research in different ways, and we just happen to be in a position to combine it into something different. So there are
I
two
didn't know about this slide.
there are two relevant studies in literature and that look at this. And again, our collaborators Sam and Lyndon are developing based vaccines that Malcolm mentioned, where you can just feed the mice and they become vaccinated, which is very promising. The downside is you have to keep feeding the mice, and they don't only become vaccinated if they've eaten enough, and if they've been infected by then you're out of luck. So it's not as permanent. And the other thing you can do is this first study is they just wanted to know, can you vaccinate the mice and would it work? So they just went out there and they trapped as many mice and vaccinated them by hand as they could. Thousands. So this was on the mainland, and they reduced the infection rate by 42% in the mice by vaccinating as many as they could. You might say, well, why not more? Well, the vaccine, when given to an individual mouse, isn't perfect. It's about 75 to 80% effective, if that, even the injected form. And then they couldn't get all the mice, because this is on the mainland. They didn't have any control over the mice coming in and out. So you had unvaccinated mice constantly coming in. But it did decrease the infection rate by 25% in the ticks, and this is a good study. They couldn't, again, control for mice moving in and out, but it was well done. So in general, I believe it. The other study was in oral bait-delivered vaccine, a different kind. That when you look at the data, it looks like it's reduced infection rates by 25% when deployed for 2 years and 76% for 5 years, which sounds really good. It was not a very good study. So I don't believe it. Not only did it not control for mouse moving in and out, but the mouse that they supposedly had vaccinated, it didn't look like they had actually been vaccinated at all. Again, this was not actually injecting the vaccine, the one we know works to 75 to 80%. We have good data on that one. This is one where they had to eat it. And if they didn't eat enough of it, then they didn't become immune enough. And when they checked for antibody levels in the mice, it looked like they weren't particularly immune, the vast majority of the mice. So how on earth are you getting data like this if your individual mice don't look like they're immune? It doesn't make any sense. Also they had serious issues with proper controls. That is, some of their controls just went way up, some of them went way down. So it looks like they just happened to start the study at a point when the initial infection rate was unusually high in a natural variability, and then it just went way down. So one of the things that we would need to be careful of on the uninhabited island trial would be initially, while we're breeding up the mice, monitoring the infection rate on that island to see how much of natural variability it is. Because we don't want a case like this where we choose it at the wrong point and then we mistake something that would have occurred anyway for an effect of our treatment. But this is, I'm afraid, all we really have. There's some additional data on which species are the most important reservoirs, mice and not. There's wide agreement that the mice are the most important, but how important is generally agreed to vary by site, with probably the mice being more important the closer to human habitation you get. That's really all we know.
So next question, and this is a couple of parts, so I'll spin it out and you can answer them. Nantucket has a history of introducing species which then explode in population. Rabbits, deer, and on the predator side, cats. The cats have apparently eliminated a lot of the small ground-based birds. And so how many mice, just in a general sense, are already on the island? You're talking about introducing as many as twice as many as are already here. What effect would that have on the predator populations, on the competitive species that are looking for the same food and nesting resources? What can you tell us about that?
It's a great question, and I should defer first that I am not an ecologist. I am more an evolutionary biologist. So I can give you my answer as best I understand it, but you're really going to want to go back to someone like Sam Telford to give you something more definitive. But in general, when you introduce a lot of prey, when you have a boom year in prey, then more of the prey survives because you basically get predator saturation if you really have a lot of introductions like that. So essentially the cats would be killing as many mice as they can, and there would still be even more mice. So you would have a lower predation rate of the introduced cats on most of the other things because a lot of their time would just be taken up eating introduced mice. And that could have effects on the next generation because you'd have more surviving other prey species. How big it would be is unclear. Sam has some data on fluctuations in mouse population that suggests that from year to year, not just on a over the season basis, but from a year to year basis, you have dramatic differences in how many mice there are, just based on how hard the winter is. And so, are you going to see more of a fluctuation than you would see going from a harsh winter to a mild winter? Probably not. But again, Sam is the person you're going to have to ask. It's a great question, though.
What competes with the mice for resources? So if we're introducing more mice, they're consuming more resources. What might suffer as a competitor?
So one option is you could just feed the mice. I mean, for us, that's trivial, but you don't necessarily want to do that. Probably you'd want to feed them in areas not near people's habitations, and then let the predators take care of the excess. But the answer is primarily the chipmunks and shrews. And to be fair, the chipmunks and shrews are the secondary reservoirs. So if you're additional competition with them, you're driving the secondary reservoirs down slightly. That would probably increase the effectiveness of the project. Would it harm the long-term viability of of those species? Possibly, but again, given the natural variability, depending on how harsh the winter is, it would probably not be significant. But ecology, this is all speculation.
Well, I'm not an ecologist either. That's why I'm asking the questions.
Emily, would you like to? I don't mean to put you on the spot, but
I think that Dr. Esfelt gave a very good answer, and it's very tall. Can you hear me okay? Yeah. So Dr. Esfelt gave a very good answer. I am a rodent ecologist. I mainly work with squirrels, which are also an introduced species here. And in the squirrel world, when you introduce them, you do see a slight increase in, a decrease in native squirrels. So my work was done in Ireland. There is a native squirrel species and an introduced squirrel species. The native squirrel species has declined. Here, if you're introducing more of the native mouse, I think Dr. Esfelt covered it quite well. They could compete with, we don't actually have chipmunks here, but we have the shrews and the voles. Those could find some competition from them, but I think in general, fairly trivial. However, this is worth looking into.
My main concern would be do mice compete with birds for resources? I don't know. I'm just asking. Just curious.
It's worth looking into.
And then I think the last one that I'll subject you to right now, can you talk more about the process of breeding this population of resistant mice off whatever island it is, and then getting enough of them to bring back to make a difference? Would you be using cloning technologies? Would you just be using natural reproduction? How would you be creating all these mice?
So that's a great question, and the answer is it depends a lot on what technologies turn out to work with these mice. That is, we're very, very, very good at engineering the house mouse, Mus musculus, which is a invasive species from Europe. To the extent to which all of those techniques will work on these mice is a little bit less clear. So there's a bunch of different ways you can do it. You can take a mouse embryo, you can deliver the DNA editing components in your new antibody encoding sequence. CRISPR cuts the target, inserts it. Then you take that embryo and you implant it into a pseudo-pregnant female mouse. And then she has pups, and that's that. The other way to do it is you can use a viral vector, that is just the shell of a virus, in which you put in your DNA and the editing components into it. And in this case, you take the male mice, you give them half a vasectomy, so you tie up one side, and the other side you deliver the vector, and so the DNA goes into their sperm-producing cells. And again, when you do this to a house mouse or a lab mouse, you get about 60% edited offspring. So the probable way we would need to do it would be to every generation do that to the males to try to add extra copies. Because again, the goal is you want as many copies of the antibody encoding cassette as you can get, because then the more copies you have on average, the fewer mice you need to introduce in the long run. And then every generation we would cross these males to newly trapped wild females, probably trapped here and then brought over to the mainland and mated. And then the question of where this would be done, could we do it at MIT? We're still talking with our animal facility. They're not super keen on the idea because that's an awful lot of mice. It might need to be someplace else. But we're not talking so many mice in the context of how many mice are typically raised for research purposes.
Can you give me a power of 10 how many mice we're talking about? 10,000,
Oh, so
100,000, a million, 10 million?
probably looking at 50,000 mice for Nantucket, 1,000 mice for the initial trial. So 1,000 mice is definitely something we can do at MIT. 50,000 mice, probably we need to find some alternative. But for example, Jackson Labs, which is a Maine org that supplies engineered mice in Maine, they ship out on the order of 3 million mice per year. So this is well within production capacity of humanity for
and this
making
is all
mice.
With mouse stacks, not cloning.
Yes.
Okay.
No cloning.
Yeah.
Attempting to clone a Paramiscus, the native white-footed mouse, would be a PhD project in and of itself.
Well, I don't know. That's what
Yeah.
I'm asking.
So yeah, definitely no cloning.
And so our local population of mice would vary, say, between 25,000 and 200,000 on a normal basis? Is that, I'm just doing the math backwards on
Yeah,
your
something like that. Probably as low as 25,000 after an extremely harsh winter, possibly as high as above 500,000 after a particularly good year at the end of the summer.
Okay, thank you
Yeah.
very much.
Mm-hmm.
Kevin, there was a couple questions asked online through some of the social media. Two of them were actually answered by you already about what happens if you eat genetically modified mice and what would happen in an event of to the native species that are already here. But the other question that was asked was, what effect, if any, would this project have on the tick-transmitted allergy to alpha-gal? If this project would also work on that, on lowering the incidence of that allergy.
I'm going to have to say that I will need to look into that because I can't answer that off the top of my head. I'll need to read the literature.
Fair enough.
Great. All right. Any other questions? I know we're about out of time. Yes.
I missed something. We're talking about Paramiscus here. Did you ever work with them before?
So our collaborators have active colonies. We have our lab is less than a year old, so we haven't imported Paramiscus into our animal facility yet. We're getting permission to do that now, but no one has engineered them before.
And also, when you're talking about Borrelia, are you talking about all the forms of it too? The Burgdorferi, not just Burgdorferi, but everything.
Great question. So Burgdorferi would be the only one affected by the anti-OSPE antibodies. Yes, we actually don't have an anti any of the others vaccine. We presumably could purify their OSPE protein and gamble that it would work, but
I've been seeing it less and less, and I've been seeing more action with co-infections. Even people get tularemia from tick bites.
mhm.
And it's spreading more and the vectors, the carriers there are less mice than there used to be. I came here in the 70s. The bull populations and the mice populations are way down. Norway rat is way up. And there are other things like Bartonella, Ehrlichia, and Babesia, things like that seem like are more on the rampage than Burgdorferi. But that's just
Mhm.
anyway. Thank you.
Thank you.
A couple more questions, and then we'll. I have a quick question for you. You said going forward, you're being funded by philanthropy for funding. What is the funding source for the project if it goes sideways? What if your funding dries up and you do this experiment on the uninhabited island and realize that there's a mistake, and as you said, you would have to get 10 or 20,000 mice, and we'd have to catch them on Nantucket to bring them over there. Are you going to have a plan B in the back, on the side at the same time, so if it does go sideways, we're able to introduce them quickly instead of waiting for that 2 or 3 year period for it to wash out? Number one, and number two, if it does go through town meeting for a warrant article, does that make the town liable for this whole project, or does that fall back on you and your backers that are proceeding with this forward? Because there's a lot of product liability in regard to this with the adverse effects.
It's a great question. So, question one, we absolutely would need to have enough funds set aside to dilute out the introduced gene for the uninhabited island trial. For number two, that gets down to complex legal regulations with what happens when there is FDA approval for something that goes forward. I can virtually guarantee you that Nantucket would not be liable. Whether we would is a much more open question in terms of potential things going wrong. That is something that needs to be looked into. But again, this would require FDA approval. So class action lawsuits against Merck for Vioxx, is this the sort of thing that is relevant? Potentially. But again, we would form a nonprofit to do this. So how much monetary reserves would be necessary to reverse the effects on Nantucket if need be? Like that's an open question as to what should be required there. For uninhabited island, absolutely. For the other one, again, I'm going to have to get back to you, but it's a great point to bring up.
If we're genetically modifying solely only for the Bergdorf fairy species here, because there are multiple species, over 300 species of Bergdorf fairy. So if the modification that we're doing here, is it only going to affect the Bergdorf fairy we're finding here? How does that move forward?
Good question. So in this case, the vaccination would be against the OspA protein, which is pretty well conserved against isolates around the world. There is not a lot of variation in that particular molecule. So it should work against most of them. I'm not going to say all of them because we certainly haven't sequenced everything that's out there, but it should work against most of them. But it will not work against any of the other actual species of Borrelia. It will not work against Miyamotoi or Myoni or any of the others.
Well, one more and then we'll summarize.
Hi. Thank you. I'm not a scientist. I'm a nature writer, so I'm not sure I really understand the science of this, and I apologize because I was late and you might have covered this. But I watched the PBS special on genetic roulette, and there was a concern raised that the agent that prohibits the cells from rejecting strange DNA also prohibits them from rejecting cancer, and that can be transmitted even agriculturally. And I was wondering if that's a concern in this case.
Sorry, could you repeat? There's a concern that the agent that normally rejects...
The agent that prohibits cells from rejecting alien material will also prevent the cells from rejecting alien material like cancer-causing cells.
I guess I'm not drawing a direct connection as to what exactly that could be referring to. It's, in terms of could this cause cancer in the mice, seems exceedingly unlikely. Specifically the cells that would be making these would actually be muscle cells, which are probably the least likely to become cancerous. But
good to know.
I'd be happy to look into that in more detail if you could send me the exact reference.
Yes, it's the PBS special Genetic Roulette, which concerns some possible health effects from genetically modified agricultural products. So the concern was that the increase in intestinal cancers in America was coming from the increased use of GMO crops that people were eating, and that the same mechanism that was turning off the ability to reject cancer was working in the gut as well.
Okay, I think I was confused by the latter bit. In this case, we would definitely not be putting anything into the mice that is put into any of the GMO crops on the market. Those are all transgenes introduced from another species. This is the same gene. These would be antibodies native to the mice. So you definitely should not see anything of relevant of that sort.
So it wouldn't be like a human being rejecting a kidney from someone else?
No. You should, in this case, we would know exactly which antibodies we were putting in there. We would not even be gambling. Because in some sense, immune systems can go wrong, right? Your defense system can also attack you. And to some extent, that is what goes wrong when you're infected with a pathogen such as Borrelia. But in this case, we know exactly what antibody we are putting in the mice. We know we would actually measure exactly how strongly it binds to its molecular target and doesn't bind to everything else. So this is, in some sense, what you might call an overdetermined system. We actually know exactly what we're doing to a much greater extent when it comes to the molecular level. The irony now is that we are much better with molecules than we are with really complex ecosystems. So I can tell you what the molecule will and will not do, but again, once you start scaling up to too many levels of complexity, it becomes hard to say. Beyond this, it is worth saying that, again, I am someone who works with molecules like that. That's my specialty. I am probably biased towards that kind of solution, and you should keep that in mind. I mean, everyone is biased. That is my particular bias. I think that this sort of thing is probably the smallest possible change you can make, and I am more comfortable making a change like that than I am doing something at a higher level because we don't understand the nature of the change that we're making. And that's just my bias.
Well, thank you for your
Thank you.
honesty.
Okay, let's do one more and then we'll stop.
Okay, does this type of technology, if that's the correct term, have any potential to learn more about preventing even worse things like malaria? Can it be adapted to mosquitoes and so forth?
Yeah, so can the technology be adapted to anything else? And in this case, in principle, the same approach could apply to any disease that is transmitted from an animal to people. However, this approach, again, only works because there are only so many mice here. If you wanted to apply it to a context where there is a lot more of that reservoir animal, whatever it is, then you would have to release so many of that kind of organism that it would just not be feasible. But that's where the technology gene drive comes in, because there you can release many fewer and convert the whole population. So if we do want to eradicate malaria, then something like gene drive to alter all of the mosquitoes so they can't transmit it would be probably one of the best solutions that currently looks like it's on the table. Specifically in Africa, it's very hard to imagine anything else other than a really good vaccine being developed that could allow us to actually eradicate a disease like malaria. And one of the reasons why I would be reluctant to even suggest that we might try it here is if we did and anything at all went wrong, we would jeopardize the chance to use that technology against malaria in Africa. And terrible as these diseases are here, children die every minute of malaria. And I would not want to do anything whatsoever that could potentially jeopardize a possible way to prevent that from happening.
Okay, well, thank you very much. I'm sure, Kevin, you'll take questions by email or
Absolutely.
whatever by phone. And we will continue this. Again, I like his terms. This is an open community project. It's not just in his lab. We'll keep everyone informed. He will come back. Emily's our representative on the steering committee. And we will see how it goes. And it may come a point where we decide it's not going to work. We may come a point where it's just not feasible, or as the majority of the island at some point doesn't want us to do it. It's the whole concept of this project. Open, lots of oversight. But I think it's, to me, scientifically, as a scientist, this is really quite intriguing. And thank you.
Thank you, all of you.
One more, and then we'll go.
So we are actively setting up a website to deal with it. Right now, our group's website is sculptingevolution.org. Probably the easiest way for us to put up information is there. You can see, for example, the grant that we applied to for government funding for the laboratory work. We've posted the application there. They haven't told us whether we've gotten it yet. You can similarly see basis of the technology that we're using, and we will absolutely update that. We're also building a site called Responsive Science, which has encountered some delays, Turns out it's sometimes easier to do things in the laboratory with biology than it is with information technology.
Right. So again, you can contact Kevin or Emily or myself or Sam Telford at Tufts. All very open to questions.
Sending out a notice of a meeting is much better than relying on people to find out that the meeting is happening.
We would be very happy to set up a list. Again, just contact me.
Website again?
And what's the meeting tomorrow?
Oh, that's just our regular Board of Health meeting. You're welcome to come and hear about variances of septic systems if you'd like.
Please.
It's riveting. Okay.
Okay, thank you.
Thank you all.